For nutrition in quants, what we are looking at is basically the outcomes they are actually here. Before we actually start, let's take a look at how we understand the outcomes and how we translate to the learning that is supposed to be possible between you and your notes versus understanding achieving the outcomes. First, the important words that we are trying to look out for here would be the idea of identifying and labeling the parts, tissue structure and cellular structure, making sure you know the difference between the cells and tissues of a dicotyledonous leaf. We only learn the dicotyledonous leaf, so you don't have to worry about the other type of leaf, commonly known as the monocotyledonous leaf. And in always common sense, you get the transverse section, as well as a photomicrograph with the light microscope, as well as describing, that's the second one, the importance of the features, including the distribution of the chloroplast. So we are looking at the distribution of chloroplast, looking at the guard cells. Alright, as well as your helicate mesophyll. And the third one that we are looking at the distribution will be the spongy mesophyll. Alright. And so, other than this distribution, we are also looking at the stomata and the mesophyll cells in the importance of aqueous exchange, as well as the vascular bundle involved in the transport. Alright, so we need to also know that we are trying to look at it in terms of three things. Okay, in terms of the three things that we're looking at, that will be the first one, which is the stem, the arrangement, number two, the root, and number three, of course, the arrangement of the vascular bundle in the leaf. And also to be able to state the equation in words as well as the chemical symbols. Alright, this is looking at the chemical symbols. And we are expected to describe in words the intake. We'll describe in words the intake of the carbon dioxide and the water by the plants. So in other words, this kind of questions that is actually pinpointed by this part of it, we are looking at the pathway taken by the CO2 as well as the water molecules into the plants. So we have to pay attention to how these two are actually going into the plant. The next part of the outcome that we are looking at will probably be just the state that the chlorophyll traps the light energy and converts it into chemical energy. Where many of us are looking at here is what kind of words do we actually use? The typical biological literate terms that we look at will be the trapping of the light energy and converting it into the chemical energy. So and usually if asked about the roles, this is basically like the role of chlorophyll. We can actually copy straight away from the learning outcome. And what is the purpose of doing so? This is the purpose, as we are talking about, for the formation of the carbohydrates. And to be very explicit about it, is the formation of the glucose. And the subsequent uses that we are talking about here, this part we are actually emphasizing on the fate of glucose. So of all which these things are covered inside our notes, am I right? And for all the experiments that you have read about, we are expected to investigate and discuss the effects. Do not have to memorize the steps, but you need to know how do you actually vary a few things. Changing the light intensity, keeping the rest of the factors constant, carbon dioxide concentration by using sodium hydrogen carbonate, as well as the effect of temperature on the rate of photosynthesis. And experimentally, you must be able to know how to calculate rate of photosynthesis. Alright, based experimentally and suggest steps how you can actually do it. Okay, and the next one is about discussing light intensity as factors. Light intensity, carbon dioxide concentration, graph as limiting factors on the rate of photosynthesis. Simply put, it is actually referring to the graph questions that you are looking at, whereby the factor is actually here and here is actually your rate of photosynthesis. So this question what I'm looking at is actually interpreting the graph, interpreting data and the graph. So after all I've been through about these learning outcomes, we should be very sure how we can actually go forward to um, uh, study thoroughly the topic. Look at this part carefully before you go on. For our subsequent revision, I'll only look at the important parts and the things that you probably have some issues with. Other than that, the rest of it is up to you, okay? Let's look at the first labeling through some important stuff that we want to look at. Let's we'll top the top layer of the leaf here. Sorry about it. Okay, the top layer of the leaf here, as you look here, which is the cuticle. Cuticle being the one that actually colorless and is actually waxy for reducing water loss. Alright, so it allows sunlight or light energy per se, the most important one. The light energy actually goes through here, right? The light energy goes through the cuticle and goes down into the epidermal cell and reaches the palisade mesophyll or even the spongy mesophyll cells here that we are looking at. Okay, the second one is this layer that we're looking at. Emphasize to you that this layer here is just called the palisade mesophyll. The cells are actually identified by their shapes. Alright, by their shapes. So you can either call this the palisade mesophyll layer or I call this the palisade mesophyll cells. Never leave it in the form of palisade mesophyll, which does no meaning if these two words behind are not there. As you realize that this region here that we are looking at is a bit of interesting part we're looking at here, you will see some spongy mesophyll cells. If it occurs, it's fine. It doesn't matter that it doesn't mean that your spongy mesophyll must be way below the center part of the leaf. Alright. So knowing that in mind, following that as we go on, if you go on to the subsequent layer, you will see that there is of course our spongy mesophyll layer, or I call it the cells. Alright, and knowing that there is also a very important layer here that we must not forget, which is this thing here. This is our vascular bundle. Sometimes, many a times, people call it the vein of the leaf. And labeling the region here would take a look at this part here. This will naturally be the xylem generally because of the thicker walls and the larger lumen. Thicker walls, larger lumen. Alright, and conversely, this part here, this will be actually your phloem vessel. Alright, not forgetting that the spongy mesophyll cell, because their irregular arrangement, forms a layer of your this is your layer of moisture that is being formed due to the water molecules that are moving out of the spongy mesophyll cells and you stay on the surface. Listen, it's on the surface of the mesophyll cells. Alright? Okay. Moving down a little bit more, you'll be able to see that there is this whole thing together, that we call this whole thing together formed by the gut cells. We actually call this the stomata. If you do see this is called the stomata. Singular being stoma. These cells on its own, alright, without the openings, so we call this the gut cells. Sometimes the stroma is just pointing to the hole, okay, and then you can actually just look at it, okay. And since we have been looking at some of this pathway, let me just do a simple key at the side for you. So to take a look at the key here that we're looking at, alright, the key that we are looking at here is this. Well, we have the arrow here that we are looking at just now. This was actually referring to the light energy. Alright, this is the light energy. We are now going to look at the water molecules that are actually going to enter the plant, the leaves. Alright, so how should we actually do it? Let's take a look at this now. First, water molecules actually move out from the roots. Right, and after the roots usually obviously has to be taken out from the soil. So the process that occurs here is called osmosis, and from cell to cell in the root hair cells, before it reaches the xylem vessel, Alright, so when it reaches the xylem vessel, it is actually pulled up by the transpiration pool. And from the xylem vessel, when the water reaches here, it can actually move to different cells like this. Okay, this is actually what I call the movement of the water molecules. As for the gases, which is actually quite important as well, when we are looking at this, we of course we would like to take a look at how it gets into the leaf. Looking carefully, okay, sorry, let me go back a little bit more about the water molecules. The water molecules, when it reaches the spongy mesophyll here, it may come out and form this layer as it actually exits the the spongy mesophyll to form a layer of moisture here, and as the layer of moisture, they start to evaporate and it will diffuse out of the stomata. You must take note that we do not say that the water molecules diffuse out of the stomata. Is wrong. It's actually the water vapor that has formed due to evaporation that will leave through the stoma by diffusion. Alright, and the last one we'll look at is actually the gases, which I'll use an orange color one to just illustrate this to you. Okay, so as the gases are entering, they will have to go by this way into the leaves, this way. Maybe I use a different color. This color doesn't look very prominent to us. Okay, so as the gases are entering the stoma, alright, this is the CO2, and it's always by diffusion. It will dissolve into the layer of moisture before, and then the solution will actually diffuse into the spongy mesophyll cells as well as the palisade mesophyll cells. So this last arrow here we are looking at is basically the carbon dioxide gases. We need to locate the positions of the vascular bundle, so as much as those that are in the photomicrographs. So placing them together, you realize we can actually look at it carefully. Alright, so in the leaf, generally, the xylem is usually facing the top part here, and the phloem is actually below. And in the previous part of the video, you already realized that it's actually within this portion here. Okay, and so for the stem, the stem is actually in the vascular bundle is arranged in a circular manner, with the xylem inside, facing the nearer the center, and the phloem, which is on the outside. The root, the xylem faces actually star shape as well as a X. So the xylem and the phloem is always on the outside. So laying attention to this from the actual photomicrograph, you can see that this is actually the xylem, right? And the phloem is referring to this part here. This one we call it an alternating arrangement because it is first followed by the xylem, followed by the phloem. For the leaf, we don't have an exact way of actually recording how it actually goes. For photosynthesis, we need to understand the raw materials as what we have mentioned before. Always the left hand side here, these are all the raw materials, and the right hand side being the products. What is special about the word equation is that the water actually repeats on both sides. And because you cannot cancel away water in a wave equation like this, both sides must be present. The light energy and not sunlight should be written as the condition, and the chlorophyll should be written below and not chloroplast. We do not write the name of the organelle inside the equation itself. Converting this to a chemical equation, you realize that we have not balanced the equation yet. So just remember that if you have a 6 on the right hand side, there should be a 6 here, and water to balance the 12 hydrogen here. Alright, well, we should do the oxygen here So maybe this is a 6. So I'm just drawing the trial and error as 6 to 12. Alright. And if you look carefully at this, doesn't seem very balanced, isn't it? So look at the 6 here. If you look carefully now, the water here actually doesn't need to exist, so it is actually supposed to disappear. Just remember that for each of this equation, as long as for each of the compound there is a 6, then the equation is correct. That would be an easy way to remember. How do you look at it? There's a 6 here, there's a 6 here, 6 here, and there's also a 6 here. Alright, so based on the learning outcomes I mentioned on the first part, when you describe in words, there are a few things that we need to pay attention to. So try and remember how to phrase it properly. So we usually would start with the chlorophyll role. Okay, you can talk about where you actually take the energy from in the chloroplast. So all pointers for you to take note. In the chloroplast, in the palisade, and spongy mesophyll cells. What do? They trap light energy and they convert it to chemical energy. I'm going to add a bit more here. Chemical energy, where is the chemical energy stored? Stored in glucose. Alright, but for this to take place, you need the raw materials, which you should try and need to stay where they are from as well, if you are required to. The carbon dioxide naturally is from the air, and the water taken in by the roots via osmosis. Sorry. Okay, so in general, basically this is how you can phrase the answer as for photosynthesis lah. Okay. This shell. So what we are looking at here is the process, how it happens. So first, so few things first. Water and mineral salts are taken up from the soil. So this is by osmosis, like I said. Right, and we also need this going in. The carbon dioxide will diffuse into leaf, correct, through the stomata. Alright, the exact pathway, I'm not talking about it here. So with these two asterisks, with the raw materials, you will be able to make the glucose in photosynthesis in the palisade, mainly and the spongy mesophyll cells. Okay, and this glucose, basically when it's made, there are other possibilities what will happen as well. The glucose that is made can be transported up in the phloem. If you transport it up or down, alright, so I put a strip here. So this one is actually transported in the form of sucrose. Sucrose. So don't get it wrong, alright? It's transported in the form of sucrose. Give me a while. This thing is a bit strange. Okay, so it's transported in the form of sucrose. Okay, in the phloem. We talk about the fate of glucose now, so just as a quick summary so that you understand. Okay, this is basically what happens. So fate of glucose, a number of them. Fate of glucose could be number 1. Transported in the form of sucrose. Excess glucose stored as starch. Actually, the very important one is that glucose is a substrate for aerobic respiration. Alright, to release energy for some processes like active transport. Number 4, you can combine with nitrates to form your amino acids, and these amino acids, remember, they become polypeptides. Polypeptides, they become proteins, and proteins are used to make new cells in the plant. Okay, there are some other fate of glucose which you need to cover, so I'll leave it to you. This is the main 4 that is actually easy for you to remember. Our experimental setup here we are looking at is the common setup that we use to have a plant. We have not done experiment before, but using this we can vary the 3 different setups. Pay attention that only vary 1. Okay, the rest, the other factors must be constant. You cannot have 2 variables, they are different, so the experiment then is not conclusive. So looking at this, we will just take a touch on what is it that we actually change to vary light intensity. You change this one to vary light intensity. You change distance, okay, of the ruler. Wrong phrasing, sorry. Change what? Distance of the boiling tube or the plant and the light source, okay, using ruler. So in this case, this diagram is not very good. You need to have a ruler here, so you have to make sure you draw the ruler properly. Okay, this one is for number 1. I put here, so this is for leaf number 1, is to vary the light intensity. How about changing carbon dioxide concentration? You got to change this one. This number 2. Okay, you can vary the concentration. By varying the concentration, you vary the amount, volume of the CO2, not amount, volume of CO2 inside the solution itself. So number 3, changing the temperature. How do you change the temperature? Well, there's only one way to do it actually. Usually the beaker is supposed to serve as a water bath. So what you need to do is change temperature. Alright, and then you will be able to serve leaf number 3. Take attention that if you do this experiment with a very high temperature first, you may denature all the enzymes inside the plants. If you do so, then your subsequent temperatures, the experiment will not work anymore. Essentially, you're trying to count the number of bubbles here, and so please, we have studied the sources of error for this part. You need to make sure that you understand this experiment pretty well. There may be variations here and there, but generally the principles involved in changing these 3 factors, right, is about the same. For limiting factors, we are looking at any one that actually increases if we change the rate of photosynthesis. So let's take a look at the graph on 2 parts here. For example, if I put here CO2 concentration. For this first part here, increasing the CO2 concentration will always increase the rate of photosynthesis. When you reach this point here, as it goes to a plateau, increasing the CO2 concentration does not increase the rate of photosynthesis anymore, because rate of photosynthesis, let me emphasize, if you have a value, you have to quote it, is already constant. Constant, not 0. So you have to quote the values if you see it. For graphical questions like this, right, it's very, very important that you quote the values from here. Okay, this is y-axis and this is the x-axis. Okay, so in other words, just to recall this part here, this is actually the independent variable. You must remember that, please, don't get it wrong. the one that you can fix when you do the experiment, usually it is the one that you fix, okay, at least 5 times. When you do a typical experiment for your exam, usually we fix it for 5 times. Alright, so basically the explanation for the rest of the 3 other factors here, here, and here, they are all the same. So take note, when I put a leaf, it means this is where you see it. Okay, let's do a little bit of adjustment to it. Sometimes you don't only get 1 graph, you get 2 graphs. You get 2 graphs. Okay. And the interesting thing about the 2 graphs is that you actually get a very different. Take a look at this. If I tell you here, because you're investigating carbon dioxide concentration, right? And so I need to keep the other 2 constant. So sometimes they will tell you this like this. This is the temperature. Alright, and then the light intensity, they will tell you is 40 units light intensity. Example, right? Okay. So usually they are concerned, why are they interested to raise the graph like that? You see, for this part here, right? CO2 is always limiting. But when you reach this part here, CO2 is no longer limiting. So if it's no longer limiting, our question now is this: is it the temperature limiting or is it the light intensity limiting? The only way is to increase one of the factor, either the temperature or the light intensity, while you keep the other one constant. So here, for example, if the person keeps temperature constant and constructs another experiment, and he find that when he increases the light intensity to 60 units light intensity, what do you realize? The whole graph went up. Correct? So for this part now, we can say that what is the light intensity is limiting. So sometimes when you get 2 or 3 graphs, right, you should attempt to read the graph this way, and similarly you can try to explain it based on how I actually explain it to you. It's a very important concept that everybody has to digest. Have to realize that your problems with it. You came and asked me, and based on your assignment, the limiting factor is always something pretty challenging. Alright, take your time to look through the whole set of this revision. Only after you are done, and please text me if you have anything. To keep quiet and wait for the test to come. Yeah. For the Sec 4s, when you're reading this, please, this is a good refresher, and make sure that you get everything up and going. Alright, thank you.